the next thing we're going to do, and usually what I do is, most people like to see the diseases and functions, canonical pathways, and the networks, or upstream regulators. So here's what I'm going to do with disease and function. So again, go to the disease and function page. You could take a picture of this if you wanted, that's fine, but I go down below to where all the molecules are, or genes, and I go over to this Export Data tab. Click on that, and it's the same thing. Put this on desktop, and I'm going to call this, we'll go test functions. Save. And we open up this one. Again, we have to get rid of this top column here. Make that top category bold. And usually this is what I'll do is I'll just take this and put my cursor in the corner and just copy the whole sheet. And then I go to my original one, which I closed. Oh. You can go to the second tab here and just paste all that. Boom. Make that bigger. And usually I leave it sorted by p-value. I think that's the more important thing. But again, you can do activation states, so we can format this based on, and here's what I usually do. So activation state's different than fold change. So instead of doing the colors based on red and green, so I go all the way to the bottom. Select this. Go to the top, make sure it's a number. Did I convert that? Yeah. Nope. Come on. This is the activation state. So this would be standard deviations away. You could kind of think of it standard deviations away from just random noise. So that if you have a very high value in the activation state, a very positive value, and if it's over, the software says it's over 2 standard deviations away from the mean, that what they'll do is basically the evidence to say that this pathway or function is more active completely outweighs the stuff that says it's inhibited. Does that make sense? So if I had 20 pieces of information on all these genes associated with this particular biological function, and 18 out of 20 of those say that based on the direction of their expression says that this pathway or function should be more active, then it will call it active, and it'll be a very high number, right? If it's even, if it's like half and half, right? Half of the genes, the direction it goes says it's more inhibited based on previous scientific evidence, and the other half says it's more active, then it's not going to make a call. It's going to be like a number like 0. Okay. So what we can do, and usually with this conditional formatting, what I'll do is I'll do the 3-color scale again, same kind of values for the number. 0 is my lowest. So in the lowest value here, if you look at the Partec or the IPA software, it always makes these blue. Oh, blue. Kind of this, it's not like blue, but it's kind of this kind of darker blue. Make this white again. And then the highest value, it makes it orange. And I kind of like that because it makes me think of the Broncos. Not only that, but it just kind of gets your mindset kind of after a while, IPA and my brain seem to like merge. And so I think of everything in like blues and oranges when I start talking about inhibited and activated. So it's good to kind of keep with the same format as the platform you're using, because it just makes everything a lot easier. Okay. And again, so now this makes this very easy to look at. And, I can look at all these proliferation of cells. What is that doing? Oh, it seems to be more active. Ooh, development of body axis. That looks very active. This one looks very inhibited, generation of cells. Again, it just kind of gives them a way, whoever you're dealing with, a very easy way to just see what are these pathways, functions associated with my gene list, and what are they doing. And again, you can name this function. Okay. And I'm not going to do this for all of them, but basically what I would do is I would go genes, functions, and I would go back to my original data set. And I would do this for canonical pathways. Export data. Then I would do it, my last one is going to be upstream analysis. And actually, this is the one that I always stress when I give somebody their results. I always grab my collaborator and go, okay, this last tab is huge. The most things that change in a particular system, if you're looking at, most gene expression is kind of shrapnel. It's these bombs going off. And it's the bombs that you really want to get at, and you don't really see because they kind of explode. So it's the upstream regulators that are really important in your system. And so I basically stress to them, I'm like, listen, I know that we didn't measure SOX2, but based on all the genes that we see change in your system, I think that this thing is active in your system. That it's regulated probably post-transcriptionally, it's getting phosphorylated or something. We're missing the changes at the transcript level. But I bet this is you want to concentrate on this thing. Okay. And, and that's basically your Excel spreadsheet. And that's kind of the meat and potatoes of what you're going to give, your collaborator. Where did I put that? There it is. Some kind of nice, easy way for them to just kind of go in here and look and see, That way you don't get like emails and calls like 3 times a day going, hey, Mike, what does that one gene do? And I get that a lot, even when I give them this. So the nice thing is I can always go, look at your spreadsheet, man. I just gave it to you. So it's very important that, not only do you analyze somebody's data, but you give them a way to actually interrogate it themselves without actually owning IPA. Okay? So again, the spreadsheet's key. And then one thing else I want to go over. So I give them the spreadsheet, and then I give them a PowerPoint, and I'm going to show you this. Again, I know the Partech isn't working.